Good morning, my dear students of class 6, and I am happy to welcome you all to today's botany class. What are we going to learn in today's class? Today's class is going to be about the observation of plant cells under microscope. In this lesson, you have studied about the structure of the plant cell, the detailed structure of the plant cell and the parts inside the plant cell, or every part, and also the function of each part, the detailed structure of each organelle. All we have been studying in the past few classes. We still have a few parts more which will be continuing in the next class, and also we will study the differences between plant cells and animal cells in the upcoming classes. Okay. So, in between your lesson you have a activity based study which is observation of plant cells under the microscope. How will a plant cell be seen when you see under the microscope? So, the difference I will show you at the end of this class, okay, because we see a picture of a green cell in your textbook. Actually, as I told you several times, plant cells are actually colorless. When you see with your naked eye, you do not find any color at all, but beautiful colors are given in your textbook. How do we get that color? Or when it is so difficult to see under the microscope, how come the scientists have framed a clear picture of all these structures? That all we will be learning. So, practically you will be seeing the exact structure of the cell which will appear under the microscope, and let us compare with the picture in your textbook. Okay. So, to do this experiment, so what is the purpose of this activity or the experiment? The purpose of this experiment is you are going to observe the cells of onion under a microscope, because the topic is observation of plant cell under the microscope, right. So, you are choosing an onion, and you are going to take a epidermal cell out of the onion, and then you will put under the microscope and see how it appears. Okay. So, that is the purpose. So, for doing this experiment you need a set of materials, and what are those materials? The first thing what you will need is glycerin. Okay. Glycerin you must have seen maybe in your chemistry lab, or even for household purposes, sometimes for in beauty aspects all glycerin is used. Glycerin is usually colorless and it is viscous in nature. A sticky feel you will get when you put in between your fingers. Okay. So, water we are not going to add, instead we are going to use glycerin in this experiment. So, why you need glycerin to observe a onion cell under a microscope? Because after some time, suppose if you are removing a epidermal layer of onion and then you are trying to view under a microscope, it will take some time to set the cells under the microscope. No, before that the cells will dry up. So, this glycerin will keep the cells moist. That is the purpose of glycerin. So, the next requirement here is saffron in solution. Saffron in solution is a red color liquid. Okay. It will appear like a red ink. If you have seen your teachers all using a red ink bottle, the same consistency only, it will have that saffron in solution. Okay. So, why you need this colorful solution? I have told you several times, cells are all colorless. They do not have any color at all basically. Even plant cells are colorless, and it gets a green color. The plant gets green color only because of the chlorophyll pigment. By itself the cells are colorless. Similarly, animal cells are also colorless only. Then we get this red color tissue because of the presence of blood. The hemoglobin in the blood only gives red color in the case of animal cells. Okay. So, what we are going to do while trying to observe under the microscope, we are going to add some color to the cell. Okay. We are going to make it colorful, because only then you can see with your eyes. Now, if everything is going to be transparent and colorless, you cannot, colorless in the sense, see, every time in biological terms you have to use the word colorless, do not mention as white. Okay. You have to mention the term colorless. So, cells are colorless, and you are going to add a red color stain. You call it a stain. It is not a dye. When you use in a chemistry class, you use the term dye, to impart some color to some substance. In biological terms, you have to use the word stain. Okay. So, you are going to stain the colorless cells, and for using the stain, you are not going to pour as such. You are going to use a dropper. This is called the dropper. Dropper is a common term, or else technically it is called as Pasteur pipette. Why Pasteur pipette? Because Pasteur is the man who invented this. Okay, so for adding the stain to the cell, you are going to use the dropper or the Pasteur pipette. Then forceps, never in lab you are allowed to take any specimen, let it be a plant specimen or animal specimen, you are not allowed to touch with your hands. Okay, so you are going to use forceps. Forceps is to take the specimen and place it in position, either from the watch glass to the microscopic slide, like that. Whenever you are transferring, you are going to use forceps. Then obviously you need a microscope. So I have taken during the microscope lesson, I have taken you to the lab, right? I have shown you the real microscope and explained you the parts already. If you have not watched that video, please go and watch that video and then come back to this class, otherwise you may not understand clearly. So the microscope also we need for this, and then we need a needle and a brush. Why? So this forceps, along with the forceps, if the particles are going to be so small, just with the brush, because this forceps is pointed, you cannot transfer all the substances. If it is going to be a delicate substance, then you can use a brush, just touch the specimen with the brush and then transfer to the other place wherever you want to transfer. Then cover slip. There are two important glass things what you are going to use. One is called the glass slide. Okay, so this is the stage of the microscope, no? So this is stage where you will place the specimen. So directly can you put the specimen on the stage? No, right? There will be a hole inside. Actually, through this hole, the light will enter into the specimen. So directly you cannot keep it on the hole. So what you are going to do? You are going to keep the specimen, whatever you are going to watch, with this glass slide. Okay, so take a glass slide, put the specimen on the, specimen means the plant cell what you are going to observe. How you will prepare the specimen in the next slide I will tell you. So take a glass slide, put the onion cell and put it on the stage. Okay, so before you put it on the stage, you are going to add glycerin. Number 1, add glycerin. Number 2, add the saffron solution. In this case, the stain is the saffron solution. There are many stains like methylene blue, like many colored stains you have, but in this experiment we are going to use saffron solution. So put glycerin, put saffron solution and then cover it with the cover slip. Why? Because both this glycerin as well as the cover slip is going to protect your specimen from drying. Or in case suppose you are adjusting the coarse adjustment and fine adjustment knobs, when you adjust these lens, the 3 objective lens may touch and it may crush your specimen. To protect that only you are covering it with the cover slip. That's why it is called as cover slip. And these are very small in size, and even if you press it tightly it will crush off. Okay, and this one is strong one, glass slide. And for any preparation you can use the watch glass. So these are all the requirements for this observation. So to begin with, what you are going to do, you are going to take a onion. Why you are taking onion? Because the topic is observation of onion cells under the microscope. No? So take a onion, cut it into 4. Okay, cut it into 4, and you can take one scale. Actually, what is onion? You have studied already. It is a modified leaf, right? Actually, onion is a leaf. So take one scale and then you can peel like this. You try at your home also. You take an onion and you can peel like this. You will get a very thin layer. In one scale, on the top of the scale, you can peel it. Not this side, okay? Not on the side where it is dark in color. Use the side which is colorless or white in color. You will have one side, no? From there, you try to peel a thin layer. So this will be the epidermal layer of the onion. What is this called? Epidermal layer of the onion. So this is what you are going to observe under the microscope. Okay. So now you have this epidermal layer. So place on the glass slide. I told you, you are going to use a glass slide. This is the glass slide. Place this peel epidermal layer on the glass slide and then you will add 2 solutions. What all I told you? Number 1 is saffron solution because it is colorless. See, if you see like this, nothing you can see under the microscope, just it will appear like some white paper, it will appear. Okay. So, add color, that is the saffron solution, and then what you will add? You will add glycerin solution. Why glycerin solution? Because it has to keep it wet, no? If it dries, no, it will shrink off. So thin layer, right? So it has to remain wet throughout till the end of the experiment. So cover it with glycerin, and then can you see, I don't know how clear it is, there is one small slide kept on that. This is the cover slip actually. It will be a very thin slide and this is called the cover slip. In this they have not shown it stain, but actually you have to add stain, then glycerin, and then only you are going to put the cover slip. Clear? So first you have taken the peel of the epidermal layer of the onion, put it on the glass slide, add saffron in it, add glycerin, put the cover slip, and then you are going to place under the microscope. Okay. So in that class where I have explained about the microscope, I have shown the real microscope and how to adjust all. If you have not watched the video, please watch that and then only you will understand what I am going to explain now. Okay. So now you have a slide with onion epidermal cells. What you will do? First you have to lift this up. How you will lift? If you just adjust this knob, this is called as coarse adjustment knob. What is it called? Coarse adjustment knob. Why it is called coarse adjustment? Because roughly you are going to adjust first. So in this coarse adjustment knob itself, in the center you will have one more knob, that is called fine adjustment knob. Okay. So first with the coarse adjustment knob you lift this up. Okay. These three are called objective lens. So lift the objective lens up and then put the slide here. This is called the stage of the microscope, because whatever is going to be performed, see in stage you do all activities. No? So similarly whatever is going to be viewed will be placed on the stage only in the case of microscope. That is why it is called a stage. Okay. So here you put it, put the onion peel on the stage, and then again you will lower the objective lens. There are three objective lens called 10x, 45x and 100x. 10x, 45x and 100x. So 10x means it will show 10 times bigger than the original size. Okay, x is the actual size. So 10x means the onion cells will be seen 10 times bigger than it is of the original size. 45x means the onion cells will appear 45 times bigger than the original size, and 100x means 100 times larger you can see. So generally when you see with your naked eye, can you see every cell in an onion? No, right? You can just see like a white sheet of paper. But when you put under the microscope and stain them and see individual cells, you can see. Okay. So first what you can do, you can view through 10x, then place this 10x straight above the cover slip, and then you will do fine adjustment, adjust it finely, then you can see the clear cells. Okay. So how it will appear after you see? It will appear like this. But what did you learn in your textbook? This is what you learnt, no? This is the structure of the plant cell you learnt. But what is the actual structure? This is the structure. Okay, this is only one cell. This one rectangle is this one. Okay. Why? Where are the other parts? How many parts can you see? This is the nucleus. You find circles, no? The center dark circle, this is the nucleus. Where is the nucleus here? This one is the nucleus, right? So when you observe under the laboratory compound microscope, what you have in your laboratory in our school, no, that is called as compound microscope. So with the help of compound microscope, you can see only the nucleus when it is stained. This is the nucleus. All these round circles are nucleus. Okay, many cells are displayed here. So why not other parts? Why other parts are not seen? You have to go for transmission electron microscopy to view this clear structure. Okay, which you can find in big research institutes. Very few research institutes will be having. So nucleus you have identified. What about the other parts you have identified? What you can see here in this structure, this is the one which appears under the microscope. Okay. So what else can you see? You can see cell wall and cell membrane. There are two layers here, no? The double layered cell wall you can see here. This is the cell wall. The double layered cell wall is the darkest green color you have, no? That is the cell wall, and here it is shown in lavender color, right? The purple color is seen as a double layer, that is the cell wall. Okay. Why other parts are not stained? You even have a big vacuole, no? Why you can't see vacuole? You can see nucleus which is smaller than the vacuole, but the larger vacuole, why you cannot see? Because these stains, either methylene blue or safranin, the stains what we are using in the laboratory have the property to stain only two parts of the cell. Okay, what are they? One is the nucleus and the other one is the cell wall. That's why in the structure what you see under the microscope, you can see only the nucleus and the cell wall. Okay, sometimes you can see the vacuoles also, but they are not stained, just like a big ball filled with water you can see. Maybe when you come to school, no, we'll observe this in real. I'll take you to the laboratory and show you. Okay, and also can you see some small dotted structure? It is the cytoplasm. Okay. So it is not colored, it appears as a colorless solution. Here it appears like a pale yellow in color, which is the cytoplasm. Where is the cytoplasm here? This liquid portion is the cytoplasm, right? So this one is the cytoplasm when you see under the microscope. So actually under the microscope you can see vacuoles, nucleus, cell wall, and cell membrane you cannot see because cell membrane is very thin in nature and also it does not get stained. You cannot color the cell membrane, does not absorb the stain, so you cannot see cell membrane. So for your homework, what you are going to do is you will draw the structure of a plant cell and also the microscopic view of a plant cell. This is the microscopic view of the plant cell. Okay. For your homework, draw this structure and near that structure you draw this structure also, and you are going to compare both of them and label the parts. Okay. So how you will label? Say for example, draw on the right side the microscopic view, on the left side you draw this one, and commonly you can mark the parts. Say for example, if this is the cell wall, you can mark like this, mark cell wall from the picture and mark cell wall from the microscopic view and then write as cell wall. Okay. Similarly, nucleus also, how you will mark? This is the nucleus of the picture, this is the nucleus of the microscopic view, then you can label it as nucleus. Okay. Like this you have to do your homework. And cytoplasm also you can mark, this is the cytoplasm, and here which is the cytoplasm? This is the cytoplasm. So the whatever parts is possible you can mark. You draw this and then you label the parts and send the photo to me by WhatsApp. Okay. So as I told you, all other parts, they are also colorless and they can be seen under electron microscope. That is how researchers have found that a cell can contain all these parts. Okay. So this is about your homework. So do your homework. Previous homework, what was your previous homework? It was the gender chart, no, XX and XY chart I have given you, and few students had completed. Geoprey, good, Jinsho, Amisha, Tivish and Adonia. Very good, only these many students have completed that gender chart and had sent me. What about the other students? Why you don't complete? So before your next class, you are going to complete the previous homework as well as today's homework and send me. Okay. So finish all your homework and activities, because we will be noting down your names and some marks will be allotted for your term examinations. So if you skip, your marks will be deducted. Okay. So please finish your daily homework and activities. For today's homework, as I told you, draw the plant cell. I need two images. One is the picture given in your book and one is from today's class where you see the microscopic view of the plant cell. So, draw both of them, label the parts commonly. One label I should have and it should be marked with both the diagrams. Okay. So, if you write a cell ball, right cell ball and right side and left side, mark in the diagram of your textbook and diagram what I have displayed today. Okay. So, that is all for today's class, and until I meet you in your next class, you all take care and bye bye.